Good morning, everyone. Good morning, everyone, and welcome to the NRF-SAIAB Panel Discussion on Changing the Tides in Ocean Research through Innovation, Research, and Partnerships. My name is Sazinza. I'm from the University of Zululand, and I'm a proud beneficiary of the HDIs program. The National Research Foundation is celebrating a major milestone in its history as it commemorates its 25th anniversary since its establishment on the 1st of April 1999 as a statutory body, which was mandated to promote and support research in South Africa. To reflect on its contribution to the science system, the NRF and its national facilities are holding a number of events during the 2024-2025 financial year in honor of its quarter century of impact as South Africa's premier science council. So this NRF-SAIAB panel discussion forms part of this celebration of events to showcase the impact of the NRF on society under the theme Celebrating Innovation through Research and Partnerships. This panel discussion will unpack the NRF-SAIAB's Joint Marine Laboratories program, the JML, in advancing marine research. The principal investigators of the JML program will discuss how 4 historically disadvantaged institutions are transforming the South African ocean research space through the DSI NRF-SAIAB funded JML program. So to give us context and background about how the Joint Marine Labs programs come about is Mr. Gart van Heerden. Gart is a transformation manager at NRF-SAIAB. He leads the human capital development and transformation initiatives at NRF-SAIAB, such as the HDIs Development Program and the recently launched JML program. Gart has been of service to the NRF-SAIAB Institute for 21 years, and I think it's important to highlight that the NRF itself has been around for 25 years, and Gart has serviced the NRF-SAIAB for 21 years, which is a significant milestone. He will give an overview of the HDIs and JML programs. So Gart, if you're ready, over to you. Thank you.
Thank you, Sazi. Lucky, can I share my presentation? Okay. Thanks. Yeah, thanks, Sazi, and thanks to everyone that's connecting and attending. Yes, as Sazi mentioned, I've been around for 21 years with the NRF, but the first stint was as an IT administrator. So I started with the HDIs program in 2012. So the HDIs and Joint Marine Labs program is really a partnership between the DSI and the national facility as SAIAB. Partnerships with historically 4 historically disadvantaged universities: Port Hare University, Walter Sisulu University, the University of Zululand, and also the University of the Western Cape. Okay, I'm just trying to advance the slide. Okay. Now, just a bit of background. The strategic initiative was launched after the 2007 ASAP open call failed to achieve any transformation. And we realized that the transformation actually needs a dedicated program, and it wasn't going to come easy, because researchers on the open call accepted the terms and conditions, but then later on, when it came to taking students onto the call, researchers threw their hands up and said, they can't find any students, any black students or female students, and we realized that that transformation is really a mindset, and that you can't force it where people don't have this transformation mindset. We then decided to, instead of setting transformation targets in the open call, we would support transformation where it happened. In other words, where people were already working towards transformation, and people working with people with a transformation mindset. With the result is the next call was then split, where 2/3 of the funding went to the open call, and 1/3 of the funding was ring-fenced for researchers at historically disadvantaged universities. Now, the aim of the program was to develop marine science with a broader base of Black and female marine science researchers at historically disadvantaged universities. And currently, the Putlisa program is supporting 19 researchers across 4 HDIs, supervising 63 postgrad students at the moment. This number is down from the 2020 number, or the pre-COVID numbers, where we were supporting over 100 postgrad students. Why a dedicated program? Well, we realized that uptake of marine science by students from designated groups and turning these people into competitive researchers was a slow process. Also, the importance of marine science from a socioeconomic, biodiversity, and conservation perspective. Okay, also marine science is a key area of DSI and features heavily in Operation Phakisa. And HDIs showed the greatest potential for growth in research. Here's our numbers. Since inception in 2012. So the program made quite a difference at historically disadvantaged universities in terms of their postgrad school in marine science. So honors labs, master's labs changed significantly at UniZulu, at Forte University, and the other 2 universities. Also, we were affected by COVID. Mostly the master's group in green, where master's students struggled to finish during and after COVID, but we managed to address that after COVID. So the model that we follow is basically one where we support the researcher in growing their postgraduate research school, growing the number of postgrads in the group, and also helping them to publish what they research. So it's always been on the Putlisa side, which started in 2012. We provided access to national facility research equipment to researchers. We helped with professional development by sending researchers to conferences, to workshops, providing funding for the students through the HICD office at the NRF, and also providing research funding for the student projects. Then, also incidentally, at the SAIAB 50th anniversary, we invited Dr. Danny Adams, and he then he promised us funding to take this Putlisa idea further and beyond funding, and also provide research equipment, capital equipment. So the Joint Marine Labs, we provide a research infrastructure, but also the aim is to grow technical skills around the labs, to grow lab management skills, and the PIs are then incorporated as honorary research associates of SAIAB. So that is the model that we follow. And in developing the Joint Marine Labs, we've coined the phrase taking excellence to the people, because we always used to bring people from the historically disadvantaged universities, bring them to SAIAB, bring them to the national facility where they used our research platforms. But now we reverse the roles in that we place these labs at the universities. So in 2020, DSI provided this money to develop 4 laboratories and to provide state-of-the-art marine research infrastructure to researchers at the universities, but also to twin the expertise of each of these universities with expertise of SAIAB. So the DSI NRF Joint Marine Labs program aims to address key marine social and economic opportunities and challenges facing the country. So the 4 laboratories are at the University of Fort Hare is Marine Biodiscovery and Bioeconomy Laboratory. At the University of Western Cape, Marine Microplastics Laboratory. At UniZulu, it's our Eco-Toxicology Lab, and at Walter Sisulu University, Coastal Livelihood and Ecology. So the initial role of SAIAB DSI was to provide the initial capital grant for these labs. The labs are managed by a management committee, which also includes an ASAP and a DSI representative, and these committees meet twice a year. It's integrated into ASAP Puhlisa and other marine platforms like SAIAB, SAION, and Smackery, and also, as I mentioned, the integration of the PIs into SAIAB as honorary research associates. Benefits of this approach is that young Black academics are mentored into lab management in terms of occupational health and safety, supply chain management, etc. We're drawing on SAIAB's decade-long experience in infrastructure provision. And the infrastructure is embedded into an established network, which is the ASAP Puhlisa program. Infrastructure is also combined with other ASAP Puhlisa components, for example, bursaries to students and running costs. So, yeah, thanks. This is the first phase of the program. The next phase would be refining what we have achieved, and getting all the committees and all the governance working properly before we go back to the DSI to ask for funding for the second phase. So yeah, without further ado, I'll give over to the PIs to give us specific presentations. Thank you. Thanks, Sazi.
Okay, thank you, God. Thank you for the comprehensive overview of the ASAP Puhlisa program and the JML program as well. Before we go to the principal investigators and their presentation, I just want to encourage the audience to take down questions or notes, any questions they have for each of the presenters, and they'll have an opportunity at the end of all the presentations when I hand over to Prof. Francesca to ask questions or feedback or any sort of engagement that they want to have at a later stage. Our first speaker is Dr. Timbinkosi Klaza. is a lecturer from the Biological and Environmental Sciences Department at Walter Sisulu University, and is a principal investigator for the Rural Coastal Sustainability Laboratory. Dr. Dlaza, if you are ready, please go ahead. Thank you.
Thank you so much, Sazi. I'm not sure if I should switch on my camera. Okay. Thank you so much, Sazi, for the wonderful introduction, and good morning, colleagues and everyone in the audience. As Garth has just mentioned, WUSU is one of the beneficiaries from the program of the Joint Marine Laboratories, and we just launched our laboratory about 5 months ago. So we are the youngest laboratory out of the 4 that was just launched on the 24th of January. May we go to the next slide, Lucky? Walter Sisulu University, by its nature, the way it is positioned is such that it is found in rural areas. Now that brings a lot of challenges with it in terms of marine sciences. On top of that, then the university is within very poor communities, underdeveloped, lack of infrastructure in terms of like things like roads, clinics, hospitals, market, as well as, in terms of employment opportunities. So it's a very isolated space that we operate in. As you will see then in the graph or in the map that is shown there, there are those yellow dotted dots on the left of the South African map. Basically, that map was generated by CSIR when they were looking in terms of vulnerability within the Eastern Cape province. And then what they identified was that the easternmost part of the Eastern Cape province, which is basically the Wild Coast or the former Transkei, is the area that is prone to vulnerability in terms of the society being isolated from the markets, the society not having access to job opportunities, and the society being isolated even from the network of the country, be it market networks, be it academic networks. So as a result, then with Walter Sisulu University being then set in such a space, it basically means then that we then interact a lot with communities that are quite isolated from the rest of the country. May we go to the next slide? Like you can see even in the pictures, like the areas that we travel on, quite a lot of underdeveloped roads, and yeah. So what that means then in the absence of job opportunities, in the absence of academic integration, it means then that the society then in this area then depends a lot on marine resources. But of course, they cannot exploit the offshore resources because of lack of equipment to access such space. As a result, then they focus a lot on the shallow marine coastal environments where they harvest different organisms, but at the same time, they utilize the ocean for spiritual purposes, be it for cleansing ceremonies, be it for spiritual communication. And then, of course, they utilize, the baseline is that they depend a lot heavily on the shallow marine coastal environments for their own livelihoods. May we go to this next slide, Lucky? Right, so what then that means is that we need to change our approach. We need to change the relationship then with the ocean, because what happens is that their reliance on the community reliance on the shallow marine environments puts a lot of pressure on the rocky shores, puts a lot of pressure on the estuaries, and therefore then kind of threatens the species that are found in those areas. Therefore, then following the United Nations Ocean Decade for Africa, then it means then that something needs to happen. We cannot take it as business as usual. But then for that to happen, for the relationship of the communities with the ocean to change, the community needs to be part of the ocean's economy. They need to feel like they are part of the changes in the tides in relation to the benefits from the ocean. And for them to do that, it means then that they should be in a position then to take whatever they get from the ocean, and then it should be then able to go through the food value chain, like from the rocky shores to the market, and then to the processing plants, and then of course to the waste management afterwards. Whatever they've produced or whatever they've caught and whatever they've sold, they should be in a position then to manage so that they not only put pressure on the ocean, they not only reduce the pressure on the ocean, but they also don't then pollute with whatever they've taken from the ocean. And that was the basis of our establishment of this JML at WUSU, where we look now at coastal, rural, sustainable utilization of the resources. May you go to the next slide, Lucky? So then, us being in a rural environment, as you may have seen, then the pictures showing the roads that we travel on, long distances, untarred roads, summer periods when there's lots of rains, easily getting stuck. And then knowing Transkei or the Wild Coast as one of the areas, in fact, is the area that has got the highest number of estuaries in South Africa, meaning that there's a lot of research that needs to be done in the estuaries within this area. And we are the closest people then that should be then tapping into that space. Then one of the requirements that we needed to have for our joint marine laboratory was to have a vehicle that will be able then to take us to the various environments and not get stuck or stranded as we go there. And then, of course, when we want to do spatial connectivity research, where we want to, for instance, look at how then the ocean then is connected to the estuary and connected to the freshwater environment, then we need to be able to navigate through those spaces. So that was the first component of our joint marine laboratory. May we go to the next slide, Lucky? And then the second component of the joint marine laboratory at Walter Sisulu University was that the university invested a lot of money in terms of constructing a brand new laboratory, and then to ensure that the equipment that we will be getting through the Joint Marine Laboratories program is well catered for. And therefore then we managed then to focus to get microscopes that will help us in terms of taxonomy, in terms of biodiscovery, in terms of species, identification at a very, at a very high resolution. So the microscopes that we got an inverted microscope, we got a dissecting microscope, and then we also got a light microscope that have got, of course, infrared and DIC, capabilities and very high resolution, then so that we can be able then to identify whatever microbial organisms that will be there to whatever level that we can be able to identify. Thank you. Let's go to the next slide, Lucky. And then the third component of the laboratory then was for the ecophysiology and histology component of research. Because remember that if you've got a community that depends a lot on coastal resources, meaning that they harvest these resources and then directly take them to their ports, there's no testing of how healthy these products are. And, diseases are easily transferred from up the food chain. And then it meant then that we needed to have temperature control rooms in order for us to simulate experiments in terms of climate change. How will climate change then influence these species going forward? What happens if, for instance, we've got 2 degrees increase in the seawater surfaces? What happens if we get a lot of dilution, as we expect, then of course with these unplanned rainfalls that happen? And then what happens to the tissue itself? You know, it's all good and well to see that if you expose animals maybe to certain environmental conditions, they die. But it's important to also see what damage, maybe sometimes they might not die, they might survive, but their survival might be limited to them maybe not reproducing. They might just be there to surviving. So it's important to also look at cellular structure, look at tissue structure, so that you can see the damages that have happened on the animal or the plant itself. Can we go to the next slide, please, Lucky? And then the fourth component of the laboratory, of course, is the aquaculture. If, for instance, you remember, in the Eastern Cape research agenda, one of the agendas that were listed there is that there is a need for us to determine, for instance, the optimum conditions for the species, for our marine species to survive in captive conditions. So this particular section then deals specifically with that to check which new species can be introduced into the South African economy, but also to check if the species that are there, that are being harvested at high concentrations in the rocky shores, can they be then taken to the laboratory and kept captive, such that we can increase their biomass. So that's what we've already started at the moment. In the laboratory, we've got blacktail, which is one of the species that is heavily abundant or heavily caught by the fishermen on the rocky shores. So we've already collected some juveniles there from the rocky shores and put them in the laboratory just to check if they will be able to survive under these conditions. We've taken mullet, juvenile mullets, and we've also taken juvenile tilapia from the estuary, and we're experimenting with those 3 to see if they can survive or not. Can we go to the next slide, please, Lucky? So this is just a glimpse of the projects that we are going to embark on using this particular laboratory. As I said, that we need, of course, to look at spatial connectivity from a biological perspective, where we look at microbial organisms, where we look at the flora and fauna that is found there, we look at the sediments, we look at the environmental parameters. So that is one of our major projects that we will then do at the various estuaries along the coastline. And then, of course, we need to look at changes in the physiology of organisms, be it at cellular level, be it at reproductive level. And then in terms of associations with the species, are we getting any new infections coming through, or are we getting new species coming into the system? May we go to the next slide, please, Lucky? Right. So in terms of partnerships, this particular laboratory currently is partnered with, remember, before I can go into the partnerships, remember that the Department of Forestry, Fisheries and Environment, what they did, they gave out these fishing rights in order to establish the small-scale fishing cooperatives. And then the Wild Coast, or the Transkei region, got 57 of those cooperatives. And then what we then did as a university, we said, okay, fine, let's adopt some of these cooperatives so that we can assist them in, in terms of compliance, in terms of governance, in terms of finding the target species that they need to farm or they need to use. And then, of course, in terms of market research, in terms of value chain. So in so doing, then we forged partnerships, for instance, within the university through the Risk and Vulnerability Science Center of the university. And then through that, then we formed partnerships, of course, through with the entities such as your municipalities, your local municipalities, your KSDs, your Ports and Johns, and then of course with UNISA, so that we can be in a position to assist these small scale cooperatives to achieve their target and mandate. May we go to the next slide, please? So where we are at the moment is that as this particular laboratory, we want to excel at what we think we can do best, which is dealing with the communities, assisting where we can, learning as much as we can from the community, and then of course making the university relevant not only to the community around it, but to as many of the coastal marine communities as possible. Thank you so much.
Thank you so much, Dr. Dlaza. Our next speaker is Dr. Ntuthuko Masigane. He is a senior lecturer at the Department of Zoology at the University of Zululand and principal investigator for the Laboratory for Marine and Estuarine Ecotoxicology. Dr. Masigane, if you're ready, please go ahead.
Thank you, Sazi. Welcome everyone. So, Ntuthuko Masigane from the University of Zululand. Can you see my screen?
If you can please change the presentation view, we're seeing your notes. Thank you. Please stop share and just reshare again.
And stop sharing. Thank you all. I'm going to talk about the Marine Toxicology Lab that is based in the University of Zululand in KZN. But first, I'm going to provide the context of the marine toxicology landscape, and then go into the EcoTox lab. So, as a background, ecotoxicology is an integration of two disciplines, ecological studies and environmental toxicology. And this has helped in many ways because previously toxicology or environmental toxicology was principally based on exposing organisms to toxicants. And at times, the organisms that were used had nothing to do with a particular system of interest. So ecology has brought in that relevance to that discipline. So we try to move in that way in the discipline of ecotoxicology. Ecotoxicology is basically about assessing environmental health of particular systems, and we focus on estuarine and marine ecotoxicology. There are several ways in which these assessments can be done. But one of them that has been prevalent in literature and in other labs is a weight of evidence approach that utilizes multiple lines of evidence in defining ecosystem status. And what has been published and what everyone seems to be following is the sediment quality triad. This was designed by Peter Chapman and the team. And in this triad, basically, there are three lines of evidence that are generally used. One of them is to do with environmental chemistry. The other one is to do with ecological studies or community studies, particularly looking at benthic fauna. And the other one had to do with bioassays or exposure of standard toxicity test organisms into different contaminants or different concentrations to try and establish a cause-effect relationship Obviously, these multiple lines of evidence are not only limited to 3. There are other developments within the field that have now included bioaccumulation and other lines of evidence looking at biomarkers. In South Africa, we're not there yet, but we're moving towards that direction. And to try and also give you a scenario of what has happened within the South African context, Sean O'Donoghue published what I think was a comprehensive review of marine pollution studies in South Africa.
Dr. Masigane?
Yeah.
Sorry to disturb. There seems to be an audio issue. I don't think a lot of people can hear you properly. So if you can speak a bit louder, please.
Or it might be my microphone. I'll try and shout. So I'm going to give a bit of an overview on the South African scenario, looking at what has happened since the 60s. And this is a review that was done by Sean O'Donoghue from UKZN. And what he showed in terms of marine pollution studies that comprised ecotoxicology, indication studies, biomonitoring studies, and the likes. He showed that our activity and productivity dates back to the 60s. And in fact, I think we're doing very well in the 60s. And you see that Prof. Alec Brown from UCT published a paper in Nature looking at the effects of hydrogen sulfide on his beloved Bullia species. And since then, our productivity increased up until the early to mid 80s. And then after the early 80s, our productivity seemed to decrease sharply, which was a shame, really, if you think about the fact that one of the first standard toxicity test methods was published in 1990s by US EPA. But since then, Prof. Wepener and his team has tried to update what is happening within the South African context, and it seems like it's slowly picking up, even though there are fewer toxicologists that are still involved, particularly in the estuarine and marine ecosystems. In fact, if you look at the freshwater component, there seems to be a lot of toxicity tests that are available. Some of them standard tests using different species from different profit levels, which is ideal. And some of these species are native, some non-native, and the tests range between acute and chronic toxicity tests. But if you look at the estuarine and marine environment, there seems to be nothing available currently that is being used to assess marine pollution or ecological status. So that's where my PhD came in, trying to develop a sediment toxicity test for marine and estuarine environments using an endemic species of the invertebrates amphipoda, to try and bridge the gap of what is happening in the country. Obviously, this is at the very early stages, and we intend to continue this line of research because standard tox test methods are vital and are required by stakeholders such as Transnet and national departments of environment, fish and forestry. So the Marine Estuarine Ecotoxicology Lab is therefore very ideal in trying to bridge the gap in terms of marine estuarine ecotoxicology. And this lab was launched in UniZulu late last year in December. So it's a very young lab. I think it's the second youngest lab from the Water Zulu lab. It's still in its infancy with two major equipment that are involved or are available in the lab in terms of ICP OES for monitoring and assessing metal concentrations in estuarine and marine environments. And we also have a TOC analyzer that will help us to standardize contamination levels in these systems. There are other small equipment like microwave digesters, a couple of hot plates, micro osmometer for physiology studies within the lab, as well as sediment rollers for bioassay tests. And hopefully we'll get more equipment to try and beef up ecotoxicology lab. In terms of research program, although it is a young lab, there is a broader research program that is registered with the university's Research Ethics Committee, and basically it's about developing and benchmarking methods in the assessment of environmental health within the marine and estuarine systems. So basically looking at the 3 pillars in terms of environmental chemistry, bioassays, as well as macrobenthic ecological studies. And there are a few students that are involved in this research program under the JML lab, Marine and Estuarine Ecosocs lab. We've got a couple of PhD students, SARS and Bulgaria, looking at the research program in our Goa Bay, particularly focusing on the ecological side of things, looking at macrobenthos as well as myobenthos, which is a very scarce skill within the ecological studies. And then we've got Noah Hendricks, who's looking at microplastics in the bay, Solomon looking at organics, PCBs, pesticides, and the likes. And we've got a couple of MSc students, particularly Blessing is looking at developing a chronic sediment toxicity tests using an invertebrate amphibod. Then we've got a couple of honors students that have come in the lab to take up a few smaller research projects. Although it's in infancy, the challenge is, I think it's similar everywhere in terms of funding, which underpins a lot of operations. So dedicated lab staff is one issue, other equipment, particularly equipment that's going to be useful in assessing quantitative concentrations of organics and the likes. And these chemistry equipment are expensive. So hopefully the next phase will get some influence in terms of funding. But in terms of future direction of the lab, obviously looking at developing standard methods that are not currently available in the country. And obviously these methods need to be scientifically and legally defensible methods. And Lisa Taylor from Environment Canada lists a process of how to develop these. And these are underpinned by a lot of basic research, which is hard to fund these days. And it takes anywhere between 3 to 10 years. So we still have a lot of work to do, considering that our productivity has dropped in the last 4 or 5 decades. Eventually, once these are developed, one would like to have these accredited, and I think these will assist in terms of government, private sector partnerships to identify the needs of such services. Thank you, and I'd like to leave you with a quote from Hermans. There seems to be a conception that ecotoxicology is a larger research area done in rich countries, when in fact it is vital if we want to protect our natural resources. And I think we really do need some funding in this space. Thank you very much.
Thank you, Dr. Masigani, for the presentation. Our next speaker is Professor Anusha Rajkaran. She's an associate professor from the Department of Biodiversity and Conservation Biology at the University of the Western Cape, and she's also the principal investigator for the Laboratory for Microplastics and Coastal Research. Thank you, Prof. Anusha.
Good morning, everyone. I just want to make sure that you can hear me and see my screen.
Loud and clear. Thanks, Anusha.
Perfect. So it's actually a really great pleasure and quite humbling to be part of this panel. Noting that the NRF is 25 years old, I realized this morning that my first year at university was the NRF's first year of existence. and that the NRF has actually supported me for the last 22 years, either through postgraduate bursaries, followed by Tutuka funding, and most recently marine and coastal research grants. So the NRF has had a major impact on all aspects of my career, and that leads to today and the JML lab that is based at the University of the Western Cape, which I'm very privileged to be the lead investigator. So it's very humbling, it's a great pleasure, and it's a great challenge that I can now support the NRF's objectives in transforming the marine landscape in terms of training and increasing human capacity. So our JML lab, which is called Laboratory for Microplastics and Coastal Research, I don't think I can call it as young as Themba Nkosi's lab and Ntuk's lab, because we were launched in April 2003. So we're just over, I guess, a year old. The objectives of our lab are to establish a comprehensive data set on microplastics in the estuaries of South Africa. I think everybody who is well connected to either social media platforms or just news in general knows that the impact of plastics on all aspects of life, including being quite prevalent within our own human bodies, as well as in all ecosystems and habitats. So this particular laboratory focuses on estuaries in South Africa, and of course estuaries are the connectors between the freshwater environment and the marine environment. So we have this catchment to coast opportunity to track microplastics as they move from one area to another. We also want to establish microplastic levels in key estuarine species. So we know estuaries provide a great deal of ecosystem services, in particular to provide nursery habitats and refugia areas for all sorts of organisms that are not only important to the environment, but also important to the human communities that live around the estuaries. So knowing about the microplastic levels in these species is critical, not only to the provision of ecosystem services, but also the health of any people living around the estuaries. We also want to know a little bit about, or not a little bit, but we also want to know about the epiplastic community or the plastosphere. With plastics being so prevalent within any environment, they provide a novel habitat for organisms. So it's important for us to know what these organisms are, how quickly they are able to colonize the plastics, and what happens over time in terms of succession on those plastics. I'm just waiting for my slides to change. So in terms of innovation, this particular laboratory is going to help in the development of strategies to monitor and hopefully in the future control pollution. And when we say control pollution, I think we all try to take that with a pinch of salt. If we look at what's happening currently with all the floods that are flowing through our rivers into the estuaries, into the ocean, we know that not only is it picking up some of all the chemicals that Dr. Masikane was talking about, it's picking up the plastics, it's picking up invasive plant species, and it's pushing all of that out into the marine environment. So we need to understand how much of the pollution we can control, where are the best places within, in terms of a holistic overview, where do we need to control the pollution, and to develop strategies around that. We also hope to provide some information into how our estuaries are contributing to being nature-based solutions to climate change. So we know that within our blue carbon habitats, such as mangroves, seagrasses, and salt marshes, the sediment environment is a very important sink for carbon, which is stored there for a very long period of time. We also, with our preliminary investigations, have found that the microplastics are also finding themselves within the sediment environment. So the sediment becomes a very important sink. How does this interaction affect the provision of the ecosystem services of carbon sequestration and carbon storage? So we hope to contribute to that type of research. It will also contribute to social inclusion and to change. We know that in terms of pollution, we need to change behaviors. How can we translate our research into something that is easily understood and that changes research as well? So in terms of data collection and analysis, we are looking at how to sample appropriately for microplastics, how to create a appropriate workflow from sampling to analysis, and how we can contribute to long-term monitoring protocols that are happening around the country. And of course, we want to be responsive to our changing landscapes. With these large-scale weather events, how does that influence the flow of microplastics from our freshwater environments to the marine environments? We want to contribute to sustainable practices. We know that I think within most research programs, we have a high dependency on plastics. How can we change that? What can we do in terms of the way we sample and the way we do our research to be more sustainable? And I've put genetic research here at the bottom. So while the lab doesn't specifically do any genetic research, we still rely on advances in genetic research to understand, for example, the plastosphere, to understand which microbial groups are sitting on our plastics and how that would impact on health and ecosystem services. So in terms of our laboratory, these are some of the equipment that we were very happy and very privileged to be able to buy thanks to the JML program. So really the flagship piece of equipment is the Lumos 2 FTIR imaging microscope, which we bought from the company called Brooker. And I will go into more detail on that in the next slide. We also were able to buy a type 1 water system that provides us with high quality water in order to run our microplastics analysis. And this supports both the JML lab as well as other researchers within our department. And then we also bought secondary equipment such as sieve sets, manifold systems for filtration, as well as freezers in order to store our samples. And I think no lab can be complete without an inverter these days. So we have an inverter within the lab to try and mediate against load shedding. So our FTIR machine, an FTIR machine is able to analyze the chemical composition of materials. And we've been working to try and optimize our process from having a sample to being able to work out the different types of microplastics that we have present. So identifying the polymers of microplastics helps us to understand the potential risks associated with different types of microplastics and to develop, hopefully in the future, to develop targeted approaches for addressing their environmental impacts. And hopefully this will include developing cleanup strategies, implementing policies to reduce the use of certain types of polymers and to investigate the health impacts of microplastics in organisms that interact with them. In terms of collaboration, so within our institution, we have collaborators as well as nationally, and we haven't got any collaborators as yet internationally, but there's always scope for that. So these are a number of researchers that we're quite privileged to be able to work with. I want to highlight at the bottom here, Dr. Pumlile Kotiani Pondo, who is at the SAIAB node in Quebec, as well as Dr. Jamie Johnson. Both of these researchers have just received their PhDs within the last 6 months, and we are very happy and excited to integrate both of them into our future plans for the laboratory. Dr. Jamie Johnson in particular was critical in setting up the laboratory when she was a PhD student at the University of the Western Cape. She completed her degree and graduated in April and is now contracted to the Cape Peninsula University of Technology. But we hope in our future plans to use her expertise in our research. So in terms of societal impact and sustainability, we know that education and awareness is important. We know that if we know better, then hopefully we can do better. And we know that we have to play a major role in capacity building. We can also contribute in terms of environmental change and management by understanding ecosystem services and the impact of microplastics and other pollutants in the provision of these ecosystem services. And this is in particular to carbon storage and sink capacity. And then, of course, we need to be able to translate our research, not just for society at large, but also in terms of making policy changes. And we need to look at that carefully as the lab progresses. So I've put some of these articles here as a reminder to us that as climate change has an impact on our coastal communities and our coastal cities, we need to know where we fit within this landscape and how we can support the better management of these coastal features. So this is just some of the research that has been done in the last 2 years. So just before our laboratory was set up. So this is some of the first research that has come out on the plastosphere and the diversity of the microbial groups on the plastics that we would find sitting in different estuaries. So here we have collected data from both the plastic environment as well as the sediment environment to determine the differences between what we find on the plastics and what we find, for example, in the sediment. From this particular research, we've now moved to a master's research where at the Nahoon Estuary in East London, we have worked with Dr. Pumlile Kotiani Pondo, and we have set up cages with plastic blocks on it to look at successional changes in terms of diatoms. So diatoms are a group of phytoplankton. They're very important in terms of their contribution to primary productivity in estuaries. And we're looking at how our diatom composition changes within a month period to look at the abundance of our diatom cells as well as the diversity that we find. So this is quite interesting research that we are hoping to publish quite soon. This is some of Dr. Trishen Naidu's work at the top, which looks at the amount of microplastics that we are finding in different juvenile fish. And this goes towards looking at the quality of our habitats for the provision of the nursery function. And at the bottom here, we have some of the research that's been published by Dr. Jamie Johnson, who looked at microplastics in ecosystem engineers, such as the fiddler crabs and the mangrove mollusks, to see if the microplastics are finding their way into these very important organisms. So what is very critical is to ask the question, so what? So we have found microplastics in different organisms. So we found microplastics in the sediment. The so what aspect is critical. How do we change this? How do we alter it? How do we measure the impacts that it has on the ecosystem services? So to this end, we've put in a new grant application for the recent call of the Marine and Coastal Research Grant in the NRF to be able to support some of these questions. Of course, our research needs to be relevant, and it needs to be contributing to different levels of knowledge. So this particular lab has the capacity to contribute to the Sustainable Development Goals, particularly clean water and sanitation, climate action, life above water, life on land, life below water, life on land, as well as partnerships on goals. A lot of our works are taking place in mangrove forests, and of course our mangrove forests and salt marshes straddle the area between life on land and life below water. And in particular for life below water, the Ocean Emergency has identified plastic pollution as a major driver of change. In terms of capacity building, I've already mentioned some of the work done by Jamie Johnson. In terms of education, so Mrs. Rosemary Eager, who I think is on this call, is a PhD student who's working on microplastics in the estuaries around the Western Cape. And part of her PhD is translating the science into information that stimulates environmental education. And she'll be working very closely with particular schools, looking at how we can integrate our research into curriculum. We also have a new master's student, Amisha Canterbury, who's going to be working with Two Oceans Aquarium, and we're going to try to both look at how microplastics have influenced turtle hatchlings along the coastline, and also take that information and translate it into a display that can be used both at Two Oceans as well as the Science Center here in Cape Town. What is the best way of communicating the impact of plastics and microplastics to an organism as important as our turtles? What are the ways that we can contribute to that? In terms of challenges and future directions, I think Dr. Masikane talked about funding. Funding is obviously critical. He also talked about dedicated lab staff. So people are very critical to the sustainability of our labs. And I think something that we can all work on together is integration between the JML labs. It's very clear to me how the UWC lab can work with the University of Zululand as well as Walter Sisulu University to really make sure that our research is felt across the entire country. We would need to obviously look at student throughput rates can be a challenge, and we need to look at proper project design in terms of that. And then we need to move beyond the baseline assessments. Where do we need to go in terms of making our research even more impactful for society? And then just looking at how our research contributes to what the government departments are looking for. Recently, we sampled a number of estuaries in KwaZulu-Natal, and we did it from a mangrove forest point of view, and we communicated our findings to the Department of Fishery, Forestries, and the Environment. And of course, our particular laboratory will contribute to their concerns about health and the concerns about collecting of different marine organisms for consumption. And it also contributes to their targets on maintaining biodiversity in estuarine and marine ecosystems. So the laboratory can help different government departments in trying to achieve their objectives. And that's everything that I wanted to communicate. So once again, thank you very much, and looking forward to the panel discussion.
Thank you so much, Prof. Anusha. Our next speaker, which is the last speaker for this session, is Dr. Ntombekaya Ngumla. She's a senior laboratory technician for biochemistry and microbiology at the University of Fort Hare. She's working with the principal investigator for the Bioeconomy and Biodiscovery Laboratory, Prof. Graham Bradley. Over to you, Dr. Ngumla.
Good morning, everyone. Thank you very much for this opportunity. Am I audible? Are my slides visible?
You're audible and the slides are visible. Thank you.
Thank you very much for this opportunity. I'm going to be telling you more about the JML labs that are at the University of Fort Hare, one of them being the Bioeconomy and Biodiscovery Laboratory under the principal investigator, Professor Graham Bradley from the Department of Biochemistry and Microbiology. This JML lab is situated at the University of Fort Hare in the Botany Building and currently has 2 PhD students, 4 MSc students, and 2 honors students that are working on their research project, which is on the identification of red algae species with the bioactive compounds that have the anti-cancer and the anti-diabetic activity that can be used in the drug development for the treatment as therapies for the treatment of cancer and diabetes. The lab focuses on the identification of novel marine organisms such as red algae with anti-cancer and anti-diabetic compounds, which are bioactive compounds such as the polysaccharides, phenolic pigment minerals, just to mention a few, that can not only be used for the development of drugs for these diseases, they can also be used in the cosmetic manufacturing companies, in the moisturizing, in the production of moisturizers, and also in the incorporation of antioxidants and the gelling effect on these moisturizers. These bioactive compounds that are extracted from this lab can also be used for the industrial use. For example, agar, which is extracted, which is a bioactive compound that is extracted from red algae, Gelidium praetorius, which is currently being harvested across the Wild Coast for import in China for the production of agar, which is used as a media for bacterial growth in molecular biology. It uses the standard procedures to identify the novel species and these novel compounds. As shown in the slides, the red algae, different red algae species are collected or are harvested across the East Coast, and they are cleaned, and DNA is extracted from these red algae species. And they are identified by a process known as DNA barcoding, whereby the extracted DNA is amplified using polymerase chain reaction, and the amplicons are sent for sequences in order to create the libraries for these algae species. The identified algae species or the bioactive compound that have been extracted can also be purified and be tested for their cytotoxicity on the cancer cells as well as on diabetes cells. And then they can further be identified using the bioinformatics analysis and also using the chromatographic techniques for their purity. The University of Fort Hare works together or in collaboration with several institutions. These include Nelson Mandela University for their tissue culture facility for testing these bioactive compounds on the activity on the diabetic cancer, on the diabetic cell lines, as well as for working with Rose University for testing these bioactive compounds in their tissue culture facility for their cytotoxicity on the cancer cell lines. It further works together with SAIAB for DNA sequencing, with Durham University for bioinformatics analysis, which is in an international university, and with Stellenbosch University for DNA and genome sequencing. On the slides are the pictures of the students from the University of Fort Hare that have visited these different institutions. The challenges of this type of research is the cost of research. The equipment and the consumables that are used for this type of research is very expensive. And yes, funding is there, but sometimes funding that is provided is not enough to cover the cost of everything, including accommodation and traveling for this type of research. The other challenge is making the pilot studies commercially available to the market. Yes, there is support via TIA, but the process is very, very long. The benefits of these collaborations, the students having access to the facilities that the University of Forte does not have, such as the tissue culture labs and the DNA sequencing equipment that are in other institutions and the Forte does not have. The other benefits of this cross-institutional and cross-international collaboration is that the students are exposed to international research. We have recently had one of our students doing their research in Durham University overseas, performing their bioinformatics analysis of their PhD study. Also, one of the other benefit is the establishment of research collaborations for the students for their career development, such as the students being able to collaborate with the researchers from overseas or the international researchers in the publishing of articles. The focus of this JML lab is to find the bioactive compounds that can be extracted from red algae species through simple processes, as well as informing the indigenous, giving the indigenous knowledge to the developing local communities along the wild coast that are currently harvesting red algae for export, as to say, this red algae that is being harvested here can be used here in South Africa for the production of agar, which can now be available in South Africa and be purchased at a cheaper price, which thereby create more job opportunities for the communities that are harvesting this red algae. The capacity and education, the students are given opportunities to attend the training workshops to sharpen their bench work and analytical skills. These include the workshops that are organized by SAIAB, such as the Introduction to Molecular Biology that is hosted by Dr. Macha. We've also had two of our PhD students attending a semester program in which they were trained on disciplines such as the oceanography and system climate array. We've also had one of our PhD students being hosted at the Central Analytical Facility for DNA sequencing for the ion tolerant and Oxford Nanopure training sequencing. The two images on the slides show when our students attended the semester program, and the other one is the facility, central facility that one of our students hosted one of our students for ion tolerant and Oxford Nanopure sequencing training. This JML lab, the Biodiscovery and Bioeconomy lab, has graduated 2 honors students and 8 MSc students. The second JML lab is the Forte Aquaculture Research Unit that is under the principal investigator, Prof. Nell Vine, from the Department of Zoology and Entomology. Prof. Vine is working with in collaboration with the commercial farms together with his students, and this assists the students in gaining the invaluable experience working in the aquaculture sector. This increases the employment opportunities as 3 of the JML graduates are now working in the sector. In terms of sustainable and social impact, this research farm helps in this JML lab helps in the reduction of the fish meal used in yellowtail kingfish culture. It also helps in reducing the waste production in the abalone farming, and it also helps in abalone drenching in the rural areas. The pictures here are on the slides are of the students doing their research in these research farms. The FHARU outputs from 2020 to 2024. Prof. Vine has graduated 2 honors students, 4 master students, 1 doctoral student, and has 2 honors students, 2 master students, and 2 doctoral students currently doing the research in this research unit. He has published 7 publications, of which one of them is a review article, and has presented in 16 conferences. Thank you very much.
I'd like to thank all the panelists for their presentations. They're very comprehensive and informative. We're going to move on to the panel discussion, and I just want to highlight that the panel discussion today is hosted not only to celebrate the NRF 25 years of innovation through research and partnerships, but to coincide with World Ocean Day as well, which is an international day that takes place annually on the 8th of June to catalyze collective action for a healthy ocean and a stable climate. And NRF-SAIAB is positioned as an innovative leader in ocean research to develop the biodiversity economy and the blue economy. Leading the panel discussion is going to be Professor Francesca Pori. She is a senior scientist and environmental ambassador at NRF-SAIAB. Over to you, Prof. Pori.
Thanks so much, Sazi. I think you've said enough of how wow about this superb collage of presentations and of course underlying what these labs can do. While I was listening to these talks, I felt like I want to be a postgraduate all over again and make use of all of these opportunities. So students out there, I hope you realize about these opportunities and you are grateful for this. These laboratories, I think they're really changing the tides of science in South Africa. I've seen already excitement among some of the stakeholders. I saw Nwabisa from SAIMI leaving messages, and Stephanie from the NRF. I see there is a representation from Transnet. So without further ado, because we are running a little bit of out of time, a little late, I would like to start with some of there were a couple of questions. I think people can unmute. There was an anonymous in a Q&A. Very happy to take the question or just to read it aloud. But please, if there's any questions from the audience, raise your hand. There was one in the chat as well. So if you want to raise your hand and ask the questions. Otherwise, I've got a few points that I'm excited I would like to discuss with all of the presenters of the JLMs. Thank you. Any questions out there? Is the anonymous attendee wanted to reveal and ask, or can I just maybe read? I think that question perhaps was more directed at Garth. Okay, let's see. There's another. No? Okay. I think I'm going to read the first one, which was actually in order of appearance, was the first question. Morning. I think this question is mostly directed at Garth, but I think panelists can also intervene. How were the themes of the JLMs of the project identified? Was it guided by existing interest of the researchers at the relevant university? And how does this intersect with the research priorities at the national level? I don't know if Garth wants to give a go or if anyone else wants to. Yeah, Garth.
Yes, okay. Yeah, thank you. Yeah, correct. I mean, we did build the laboratories on existing expertise at the university. But yeah, but I mean, it became clear through the presentations what that all the labs are aligned with national priorities. I can ask each of the PIs maybe to elaborate on their own labs. Temi Nkosi.
Thank you, Garth. I think maybe just to quickly go through the question, it will be something that it was going to be impossible for Walter Sisulu University, for instance, to do research that is beyond our capacity. So the JML had to be aligned, of course, with the priorities of the university first, it being a rural university, an African university, a university that needs to have an impact to the society. Then the JML had to be aligned with that, and automatically that was aligned with the priorities of the National Research Foundation. Thank you.
If I can add also onto that, and then I'll let Anusha go. I think when you think about I agree with Thembinkosi. You needed to strategically identify the expertise already available at the university, but also what I've seen, especially today, being collated all together, all of the research that is done, all of the questions that are done, all of the motifs behind, all of the structure of this research is, I think, completely aligned with the ties of the 2030 DSI strategies: transformation, innovation, excellence, and sustainability. So I think these 4 elements, I can see them appearing very strongly in throughout the 4 laboratories. Different ways, some will highlight something more than others, but I think they're all fitting superbly, I would say, into this frame. I think Anusha wanted maybe to add something. I'm not sure. Otherwise, we've got a question from Tufik.
I just wanted, yes, sorry, Francesca. I just wanted to add, you know, during the process of applying and getting access to funds, we did have to motivate, you know, how does the labs contribute to the SDGs? How does it contribute to the UN Decade of Ocean Sciences, to the Marine and Antarctic Research Strategy? As we do with any NRF grant application, you have to provide the national strategies and the national alignment. And I think we all worked really hard to make sure that we aligned with those. Thank you.
Thank you so much. I think before we go on to Tufik, there was another question. Sorry, Tufik, I'm coming, but I think there was a question originally aimed to still Thembinkosi. I can read again, but it was basically linking to Thembinkosi. Your research really links to engagement with the rural communities. So one of the question was, have you engaged through program of awareness with the community?
As I had highlighted, thank you, Francesca, as I had highlighted in the presentation that there are specific, there are specific small-scale fishing cooperatives that we have adopted as a university at the moment. In assistance, of course, in collaboration with the Risk and Vulnerability Science Center. So what we have done already, for us to be able to see how we can assist them, we needed to, of course, to go to them. So the first thing we did was to invite all of them to the university. I think we had about 15, if not 16 cooperatives that came through. And then, of course, they highlighted their issues, the challenges that they face. And then from there, we then narrowed it down and said, okay, fine, let's adopt a specific group that we found to be slightly ahead of the other groups so that we can start working on them. And then the others will then come as, you know, as we'll use this current cohort as some sort of, you know, a guinea pig, to put it in simple terms. So what we basically did then, we identified, for instance, that they've got issues in terms of cooperative governance. They were not equipped in that space. They're not equipped in terms of financial literacy and management. As recent as on the 17th of May, in collaboration with the Nyandeni municipality, we were with the community whereby there were fishing gear that was given out to the community. So we actually actively involved with the communities almost not on a daily basis, but as frequently as we can. Thank you.
I think I'm going to go to Tufik. I promised Tufik I'm coming to you just now, but I think there was a point that was linking directly to the very point that you made, and because I appreciate the really participatory action with the engagement, full engagement with the community. What's behind, what is beyond that? So it's good that you engage with the community, but at some stage you also might have to enter in agreements and also what are they coming out with? So if you're thinking about the real impact, the afterwards impact, there are of course those type of legals, if you want to put it that way, type of engagement that one has to consider, like benefit sharing considerations, something like that. Have you thought about that process as well? Can you comment quickly on that before I go to Tufik, promise?
Okay. I think maybe what I can say regarding that is that Maybe I've just given a slight information, but there's more that we are doing at the moment. Self-reliance is the ultimate goal, because remember, these cooperatives have got 15 years to exist, and then of course after 15 years they must be independent. They must be in a process whereby they are business entities on their own. So in the process, then we're going to work hand in glove with them to make sure that should we not reach the ultimate goal of making them independent cooperatives, then we'll work towards, of course, getting to that goal. As you've seen in the last slide, for instance, there is a picture of us being involved in a muscle rehabilitation process. That was done, for instance, at Hamburg, which is not within the Wild Coast, but because we were then requested to come as a university and assist there through the WWF program. So the long-term goals are there. The reality is that we want people to be self-reliant, and we feel that we'll achieve that goal if we keep working with small cooperatives one at a time, and then eventually they'll be independent. Thank you.
Thanks so much, Thembinkosi. Very excited about the Hamburg, because I know that I directed people to you, and I'm so glad that that project has taken off. Tawfiq, please, finally, I'm letting you speak as well. Can you just unmute and ask your question? Thank you very much for your patience.
Thank you very much, Francisca. No, I just wanted to make a comment, and this is directed to Anusa as well. I want to say as well that, I mean, this is a very good initiative to have all these different labs and stuff and whatever as well. Anusa, I just want to say that the Department of Forestry, Fisheries and Environment through Oceans and Coast have also made a big investment in terms of microplastics, and to actually start a microplastics lab in Cape Town as well. I think the focus is shallow water and the deep water environment as well, and it's actually being managed by Keshney Pillay. So basically, South Africa through Keshney Pillay is also the African node as well. I think through the UN Ocean Decade Program, and they also received some funding through that as well, in terms of starting this particular microplastics lab as well. So they're doing a lot of SOPs in terms of testing various methodologies and trying to figure out whether it's the right ways and not, and actually trying to standardize it across or a global standardization, both within Africa and the North and some other countries as well. I think they're actually going to a meeting in Tunisia next week. So it would be nice for you to actually maybe get in touch with them as well, so that we can have, you know, some synergy in terms of you know, the actual microplastic labs that we're doing as well. And then in terms of pollution as well, a couple of years ago, again, Oceans and Coast invested into Walter Sisulu University, and I think we established a monitoring program for marine pollution. So I think that lab is still up running at Walter Sisulu as well. So there's another link both between microplastics pollution, DSI and DFF and the NRF platforms as well. Thank you, Francisca.
Anusha, would you like to comment on that? This is like great. We were looking for integration and partnership. So this is like excellent conversation that we're having here.
Yeah, thank you. Thank you, Tawfiq, for that. Yes, it's very important for there to be integration across different institutions that have these facilities. And I think that's something that's really lacking at the moment, because I think within the Cape Town area, even Dr. Conrad Sparks at CPUT, I think is on the call, DFFE, UWC, as well as the SAIAB node in Quebec, also has microplastics labs. And I think there does need to be maybe a committee that sits together to prioritize different aspects or different locations across the country to make sure that we can cover the entire country in terms of our microplastics research. So that is definitely something that needs to be driven, and it needs a champion. And I just don't know who the champion is and whether that champion has enough time to do everything. So yeah, so people are important in driving these initiatives. Thank you.
Thank you so much, Anusha. I'm just spinning the idea, putting the flea. I know and you are part of it. How about organizing something like national working groups? I think that might just be, there is the one on blue carbon and you're part of it. So some ideas like that could make sure that there is synergies, there are synergies rather than overlapping. And this is very, very important. I think, I don't know, I cannot see. I don't know if Tufik still has his hand
Yeah, I
up.
just wanted to say something around the Anusha, is a very good idea that we should actually have some sort of a national platform and some sort of a task group as well. You know, through the Department of Forestry, Fisheries, Environment, Oceans and Coast, we've got a working group called Ocean Sciences, and under that working group, there's a section that actually deals with microplastics, and it's actually a task group as well. And the intention of that particular thing is to actually expand it to the outside area. At this moment, it's still internally within DFFE, but they are going to expand that as well. But I mean, you seems to be like a good champion for this particular initiative as well, Anusha.
How many champions can you be, Anusha?
Only if you can clone me. Can you clone me? Can you clone every principal leader of the JML labs? Because I think we all need a clone.
Nokubonga is on the line. Then I've got two comments that I'm going to address just now. But first, Nokubonga, please, off to you.
Thanks, Francesca. I'm not sure if you can hear me.
Yeah, loud and clear.
Okay. Thanks to all the speakers. My question, I just want to direct it to Dr. Maskane. I saw among the instruments that you are having there at UniZulu, you have the ICPO-ES. I just wanted to find out, do you have it up running already? And do you also do the analysis in animal tissue samples? And is the lab accredited already? Which I'm assuming, but then I want to understand that. And I think maybe I'm asking from a selfish point of view, because I am myself interested in the metal accumulation in animal tissues and also the trophic transfer of these metals. So it would be great to come and do the analysis maybe in your lab one day, or do some collaboration work. And Anusha said a very profound statement about integrating, doing an integration of these 3 laboratories in the future. And I think it would make things easy for also students to do the analysis at a much cheaper rate, or for free if there are also collaborations that are involved. Thank you.
Thanks, Nokubonga. I think Tux would like maybe to address this. It's specifically to your equipment or the laboratory equipment.
Thanks, Francesca, and thanks for the question, Nokubonga. ICPO-ES, yes, we have it running. We haven't done much work as yet. It's difficult to pinpoint someone who's going to be hands-on the lab, but we try to circulate our technical staff there. Yes, it's running. In terms of what sort of work we're doing currently, it's just water and sediment analysis of metals, particularly in estuaries, having just done marine. In terms of animal tissues, looking at bioaccumulation studies. So far, we haven't done that. And accreditation-wise, obviously it's a long process, but there are published standard methods from USCPA and Environment Canada, CSIRO. This one can follow and give you very good data with quality assured data. Collaborations, samples, very welcome. You're very welcome to visit.
Yes.
And I've also visited Walter Sisulu's National Marine Pollution Lab. Very good collaboration potential there. And I've spoken to Prof. Nakin about that. So there's quite a lot of collaboration opportunities that one can explore. Thank you.
And while I'll keep you there in Tutuku, because there is a comment, and I'm not skipping, I'm just looking at of comments that are relevant, so I will address all of them. But Albert, I don't know, is multitasking, is in so many meetings, but he still has the time to listen to this presentation. He said, has there been any work that has been done to look at trends of bioaccumulation of microplastic and metals in our aquatic systems using museum specimens that have been collected over a long time? So like basically referring to longitudinal studies, this could be linked to assessing potential health risk and potentially also looking at projection in the future. And he says, Anusha Ntutuko, any thoughts on that?
About museum samples, I don't know if they've been reanalyzed for metal accumulation, but I know there are some reports from back in the days by CSIR, where they looked at some particularly mussels that were used for metal accumulation. And I think it ran for a little bit, and only recently that we are looking at bioaccumulation of metals and microplastics. I know Conrad Sparks has done some work on fish and others, taxa. We've had 2 students that have looked at mussels and oysters and bioaccumulation of microplastics, and one is about to complete, the other one is wrapping up his MSc. So I don't know if that answers it, but there's still a lot of work to be done, particularly in that field.
Anusha, anything to add? Maybe contamination of samples, perhaps linked to your microplastic work?
Yeah, I think it's a great opportunity, especially if it gives us a temporal view of accumulation over time. It would just come down to prioritizing which museum specimens we would use, because obviously those would destroy the specimens and they would no longer be available. So that would just be something that we would need to think about.
Thank you. Before I go back to Tufik, there was an anonymous question, and I think Ryan also linked to that. And I've also thought quite hard about that. So everything, like in many other fields, but specifically to your laboratories, how do you see that? There's a lot of, I see your laboratories, your platform as kind of well subscribed from a student point of view. You think you all seem to be engaging and training the next generation. But then what happens to these graduates? How is the job market going to take? From one side, I think Ryan also pointed up, are they taken up in the industry, or wherever are they going? And another point is the challenges that have been highlighted. Often we get infrastructure funding, but what about the people? To make a laboratory sustainable, that expertise somehow needs to be retained. Staff that has been upskilled, all of a sudden the contracts end and they without jobs. So where do you see, is this a challenge? Are you so far in your quite limited or experience, because these labs haven't gone up for too long? I think there is a Graham who's raised his hands. I don't know if he wants to answer these questions related to this point, perhaps. Yes. So Graham, please have a go, and then we can hear from the others.
Yeah. Well, as you saw today, Dr. Nkumla presented on my behalf, and the reason I've asked her to present is she is one of the success stories of this program. She was the first South African female doctoral student in biochemistry in the history of Fort Hare. And as a result, she has been developed to replace me. And yeah, we're busy handing over. So that is one of the success stories of this program. and she will be running the platform in future. The other one is Faith Masilive, is currently a PhD student. She will be graduating early next year, and she has been taken up and got a job opportunity based on her experience and research in this platform. And then Sita Nkorkwana is one of the researchers we've sent over as part of the NRF Newton Trust grant. And he went to Durham University for 3 months where he studied bioinformatics. He went to Karel van Heerden at the Central Analytical Facility at Stellenbosch and got very well trained in genome sequencing. And he will be returning, hopefully, we'll be able to retain him at Fort Hare to replace me in teaching bioinformatics at the University of Fort Hare. So the whole program is designed in order to not just train, but also to retain Black South African students in this program. Thank you.
Thanks so much, Bradley. Graham, sorry. any comment from the others panelists? Any insights? Because linked to that, if there aren't, I would just, sorry, someone unmuted, but there is also, I just looked, this thing is crazy, being a facilitator of a panel. I've got 300 things going on. But basically, Enrico also was asking what is the next step. It's very important to train, to make marine science attractive. So also, how about really reaching that awareness that we were talking about earlier to younger levels, so school learners type of awareness? Anyone would like to make a comment on that?
Yeah, I would like to just say something on that. So in my presentation, I talked about Amisha Canterbury's work. So Amisha works at the Science Centre in Cape Town and works directly with students at all levels within the school system. So there's a really great opportunity there to translate the research into something that is easily accessible to the greater society, and also, you know, we try as much as possible to interact or to connect our undergrads with people in the field and put them into the field. And we recently had a third-year student go out with the team from SAIAB to the Nahoon Estuary, worked on one of the plastic projects that we have going there. So there's a number of opportunities, and I think all of the laboratories have a great chance of making marine science exciting to the younger generation. And I think there's great momentum in that direction. Thank you.
Thanks, Anusha. I don't know, Tim Nkosi and Tooks had unmuted themselves. I don't know if they want to comment at all on any of the points that we had just discussed.
Yeah, thanks, Francesca. I just wanted to highlight that some of our graduates, they are placed in various institutions. So in terms of tracking students where they end up at. And one of them, Blessing Nkulu, is on a 2-year contract with CSIR in Durban, where they do a lot of ecotox work on a marine routine basis, and is very excited to be there. So other students do find some places and spaces. And in terms of collaborations as well, I try to collaborate particularly with CSIR because there are very few institutions that focus on ecotox work, but obviously not all of them are in marine and estuarine space, so very, very limited. And I think that's what, in terms of other steps of popularizing marine research. So our students like Saz and Mungelo are always involved in open days or annual open days at the university, where they try to attract students from high schools, particularly in the rural-based schools, talk about marine sciences as a whole. Thank you.
Thank you. Thanks. I've got a question from Anonymous. I don't know if it was the same Anonymous or another Anonymous. Are the labs open for private use, like for private consultants? I'm not fully aware about that, but if they're not, is there any scope? We're talking about how hard it is to get fundings. This is like some potential provocative question. If they're not yet open, would there be space to let them open to private consultation? Who wants to go? I don't know. You still unmute on ongoing talks. I don't know if you want to comment on that, or I know that Graham has something to say. Let's try to give space to everyone. Tux, do you want to go first?
Oh, thanks. So our EcoTalks labs is still very young. So collaboration with the private sector is very welcomed. Because it will also assist in getting the labs running. But keeping in mind that, particularly in my point of view of EcoTalks, we still want to get to a phase where we can develop standardized methods that are scientifically and legally defensible. So consultancy, I think, that is very important in terms of the EcoTalks space. So, yeah.
Thank you. Graham, do you want to have a go as well? Do you have a comment on this?
Yes, yeah. The whole purpose of developing these labs is to have these labs nationally available to anyone who wants or able to use them. But as Tux has also indicated, if it's for industrial use, it all depends on what kind of analysis needs to be done, because often in an industry, they need to have the lab certified. So it is something we will need to consider. They are available. It's part of the whole way that the labs were developed. But if we are going to make that then a standard practice for our labs, we're going to have to go the certification route, which is quite expensive and a long route. Thanks.
Thank you. I see that there was someone else who had raised their hand for a question, but I saw that there's been some correspondence between Sibusiso asking Anusha about further research into the leachates from macro and microplastic, and that was one of my question as well. And I'm trying again to play the devil's advocate there. Anusha, we've seen you have to filter specifically water because there are microplastic in the water from the tap. We've read recently there are nanoplastic in the human testes. Are we too late, or can we still do something about this? Are there solutions about it?
Yeah, it's very important to keep a positive outlook when you do this kind of research, because the question is, are we too late, is a very relevant question. I think I don't quite know how to answer it because I don't want to say yes, it's too late, because there's always something that we can do. And I think, you know, looking at alternatives, it's never too late to look at alternatives. It's never too late to change behaviors. It's never too late to educate and bring awareness. And all of those things can contribute to reducing the concentrations for the future. So yeah, it depends on your outlook on the day if it's too late or not. But the positivity is that you have to work towards change. That's
it's never too late. And I think you can, for climate mitigation comes with the change of behaviors. And if you show how grave or how serious the problem is, you have more power to action governments. And I had a question related to that, but there is Megan had her hand up. Would you like to still ask a question, Megan? Sure, we can. I still have, and I'm going to keep those 6 minutes because we were very, very good. Talking about, let's get to I've got 2 points that I'd like the panelists to answer. One is, how do you make it more effective? And I'm coming from a scientist point of view that for years has done, like, I'm interested in this question. I'm interested in understanding the mechanism. Now you need to convince government, departmental governments, and it's great that I note that there is active participation here. How do you make the research or what you find linked to, translated to government for design of policies, or in, like Graham was also, this is not about government, but also how do you get that patent? How do you get to market? How do you streamline the results, the evidence, the science evidence versus implementation of action, which is necessary, especially when we talk about climate mitigation, example of climate mitigation, or sustainability in relation to rural communities. So how do you preserve the rights? How do you make the change? How can we speed up this link? The science policy gap has been discussed.
Francesca, let me just come in so that the brilliant minds can come after. My take is that what we've done, we're working on currently, is we are working with somebody from the Western Cape who has got a company that deals with training scientists to be marine ambassadors. It's a very tricky space that we're not really sure how we're going to navigate. But the idea is that the company will be in a position to assist us to simplify our findings, to be able to, to talk to the laymen on the street about marine sciences. And also to find ways of consolidating things like terminology. For instance, if you're talking kelp in the Wild Coast, you'll find that within the Wild Coast, other guys will be calling it Imbambosi, and then others will be calling it Imbuya. And then how do you then synergize that information? Because in terms of policy, if we were to talk about, for instance, utilizing the kelps as an ecosystem in terms of trying maybe to say, can we get carbon credits using kelps? We need to be in a position first to be able to say, can we translate this kelp in layman's term? And then that translation, can it then be utilized then to talk to government and policy? Thank you.
Thanks. I don't know. I think Tufik has raised his hands and most likely is related to this conversation. So I'm going to let him in. Is it related to this, Tufik? You
it's related to the, it's related to the policy issue. Yeah.
Sure.
I mean, I've been sitting in government for many years, and one of the things that we always get wrong is policy implementation, and especially from a non-governmental institution, and how do they actually bring in some of the work that they're doing into policy as well. I mean, there's a barrier for that. But there are certain ways that we can actually break that particular barrier as well, in terms of the way that you actually get into policy as well. And there's various avenues that you can actually do that. So one of the biggest mistakes that we actually do is we do the work and we get something and we say that kelp beds are important as blue economy structures or whatever as well. And then we take that information and we shove it down, going to government and say, please put this into policy. Government is not going to do that. Okay. The government institutions need to work with individuals from the start, so that they can actually have an idea in terms of how it actually fits into the policies that they're actually doing as well. The other ways of doing it as well is to actually be part of working groups as well, where you actually present the work and then the work actually gets presented up the ladder to other working groups that then go to MINMEC and MINTEC as well. And those are some of the ways as well that we actually get into policy management. I mean, it's difficult for a scientists within government to actually make changes within policy or even contribute as well. And I'm just thinking, how difficult will it actually be then for people that's not even in government to contribute towards policy? And I think, there needs to be more courses and more talks around policy. What is policy? How do we get to make changes within policy? But how do we actually get to work with government to actually make those particular changes as well? Thanks, Francisca.
Yeah, very, very true. Also, I quite prefer, out of your two alternatives, I think they should work together, but I quite like the working groups, because sometimes problems need to be identified by the people on the ground, by the scientists, by the society, and then reported and then being. And the government needs to be convinced. So I think the power of a working group, and that's why we were referring earlier on, we need to just consider. I don't know if there's any other question. I had my last challenge for you all, or for us all rather. We talked about collaborations. We've seen how they nicely structure each. There was a slide for everyone. You got plenty on your plate. But how about, would there be a nice experiment to see, can this joint marine labs jointly get together and find an avenue in a region of interest to everyone, where you could put together, I don't know, a community of practice. We've seen Stephanie linking earlier on, that can enhance the functionality, the exposure, the credibility, the international exposure of these laboratories onto a joint project. How do you think this bet could work? Would it work? And I challenge also the SAIAB and we've got so many stakeholders here that perhaps could contribute to almost like a national avenue of a program, something like that. Unifying disciplines, unifying people.
Hi, Francisca, Stofik.
Yes.
Yeah, I mean, something like that can definitely work, but it comes down to personalities and people as well. And people need to be able to work together to actually make something better. But if different people don't work together, then we're not going to have that particular problem as well. And one of the issues that we're sitting with as well is communication. Communication is something that is very lacking as well. And I mean, we can see it in terms of how the different things are actually being developed and spurred around in terms of labs and programs and research funding and all that as well. In that we don't speak and everybody is working in silos as well. So we need to break those silos, communicate more, send information more in terms of social media and other issues so that people can actually know as well in terms of what's happening. The one thing is that the marine community in South Africa is so small, yeah, but the information don't really come through. I mean, I saw this thing about 3 nights ago on Facebook, that there's going to be some sort of a talk on the various labs and stuff and whatever as well. And then, I saw, there's an ecotoxicology lab, there's a microplastics lab. And then I looked into it in terms of, who's the people that are actually doing this. And I mean, I only found out that it was Anusha that's actually doing it today and whatever as well. But I mean, there's so many other labs. I mean, there's the Northwest University as well. There's University of South Africa as well. That's all doing all of these things. And we need to put our heads together and actually try to synergize as well. Funding, cost containment is a big issue for South Africa. In governments, whether it's DSI, whether it's Department of Forestry, Fisheries, Environment, there's cost containment. We don't have a lot of funding anymore to actually do all of these things, and we can't do all of this work in silos because it's very, very, very expensive. Thank you very much. Francisca.
Thank you. Any further comments? And while we're waiting for the comments from some of the panelists on board, Stephanie is really excited about the community of practice. So please, guys, someone needs to raise their hand and build something. And don't forget that we're talking also about the SAIAB scientists. So there's really so much potential here from one of they're asking already for a PI from you guys about this community of practice. So this is really a great opportunity. And I'm grateful that I've been part of this discussion, because I'm looking forward to see what's happening. And yes, there's another colleague, Lumco from Transnet. They also interested in further engagement. They asking if the presentation can be shared to get the contacts, to get those discussions going. But I'm waiting for some further final remarks from some of you. I'm talking too much.
I would say from my side, I think when labs are within a year of being established, I think we all need to find our feet first. And yeah, there's lots of scope for collaboration, but we also need to be realistic about what we can achieve and who we can achieve it with. So those are great ideas, and I think in the future will be, definitely be part of the 5-year or 10-year plan for the labs. But for right now, I think, once we can establish ourselves, really work on capacity building, get people to run our labs so that there can be good productivity levels, good outputs in terms of students, in terms of publications, in terms of translating the science. So yeah, I think definitely one for the future.
Thanks, Anusha. And I think you are quite right to be cautious, because you want to build the house in a with strong foundations. It links also to the final anonymous attendee comment about funding. How do you maintain that? It was in my list. You got 5 years plus maintenance grants. How do you ensure that the funding, especially if the laboratory is ongoing, is successful? You need to keep the people, keep the research going, keep the lab afloat and current and relevant to an ever also evolving type of research questions and directions. So yes, if nobody has any further comment, I'd just like to close up and to recap a little bit, to thank everybody. I've been so excited, and some keywords that have come from this discussion that I noted, the challenges of sustainability of, like we just discussed, sustainability of the laboratories, staff retention. Also, how do you stay innovative? How do you stay relevant? How do you remain agile in this very dynamic, which is part of doing innovative work, in terms of collaboration, potential unifying approaches among laboratories and other external stakeholders, and the societal impact really from all of the representation that we had today here. There is willingness. Are there working groups? Can we just find solution? Can we just move this science evidence, streamlining it to effective action? And finally, upskilling workforce from a very early level, from schools onto training of students and getting upskilling and dedicated staff. So and people sustainability. Without people, we cannot do any science, we cannot do anything. So I'd like to close it here. I'm passing back the mic to Sazi for the next part of this discussion. And I thank you all for being with us and being so engaging. Thank you from my side.
Thank you so much, Prof. I think the panel discussion was very helpful and insightful in exposing some of the activities that are currently happening in some of the labs, but also some of the endless possibilities that might come out from the interactions, the collaborations from the different principal investigators. So thank you for the discussion. The next short session is going to be, which is the final section of this event, We will hear about the role of the ASAP Puhlisa program in transforming the lives of young researchers, which I think is also very important for students like me who are still on the ASAP Puhlisa program. Also in, June, which is what we usually, celebrate as Youth Month. So this very short session, we will hear from two speakers who will share their success stories of how they benefited from the program and just a bit of some inspiration for students who are in the pipeline, but also in general, scientists who just want to do better and be better scientists. So our first speaker is Dr. Zinzisa Nokwali, a senior laboratory assistant at Walter Sisulu University, and she will tell us more about her journey with the NRF-SAIAB ASAP Puhlisa program. Dr. Zinzisa, over to you. Thank you.
Good morning, all. Yes, it's still morning. I'm actually humbled by Ulaki when he called me doctor. Hopefully it's really happening. Thank you, Lucky. I have to be a doctor now. My name is Zinzisa Nokwali from Walter Sisulu University, and I am currently a senior lab assistant / lecturer, junior lecturer, and I'm one of the success stories of Ndate Gath and the ASAP Puhlisa program. I got to be one of the beneficiaries of the program in 2014 when I was doing my honors degree. I got funding from them. You know, as a student, when you are registering at the university and you don't know what to do, you try your degree and you know what, I don't want to go to work, I want to study further. So I was actually anxious on what's going to happen to me now. So I guess I'm one of the lucky ones on that pool that I got funding from ASAP, and I did my honors. That was 2014. I got a lot of support starting from funding as well, which is tuition, and also I attended my first SAMS in Stellenbosch. It was a wow experience. I mean, I've never attended a conference before, and to meet the giants in the marine world, in the science world, where I actually, you know, my research paper was all about. I met Undate Griffiths. I met the famous McQuaid, and I met George Branch. I was like, wow. So this thing, I thought it was happening in papers and journals and all that. Now I'm seeing these people in front of me. I was like, okay, this is what I want to do. So I got all of that support from the program, and I finished my honors in record time. And my supervisors were Prof. Nakin and Prof. Francesca Pori. And they actually gave me much exposure and a lot to do and think about when it comes to the marine world, you know, the first interaction with the marine world. I mean, I grew up wanting to be a doctor, MPCHB, but in that time I was like, you know what, this is what I want to do. I'm going for this. And the support that I'm having is actually what I really love, what I have prayed for in order to actually be in this space. And I finished my honors. I was like, okay, let's go for it. I registered for my master's. And of course, the two supervisors, they took me under their wings. And I did my master's with Prof. Pori and Prof. Nakin. And it is when I actually had my eyes opened and there was an enlightenment about this field of science. And I've been to many places that I can say, if it wasn't for Puhlisa, I wouldn't have. I've been to the SA the semester. I've been to, I mean, I've never been to Cape Town before. It was my actually first time. In 2015, I did Indibano with Sion. They've supported me through many seminars and writing retreats, and it has been a pleasure to be a part of HDIs Putlisa. And then now I am actually, whilst I was listening to this seminar, you know, when Prof. Anusha was talking about microplastics, I was like, okay, this is my next step. So like, you calling me a doctor is actually coming to life. So I think I will be joining that side in looking at microplastics for my PhD. And yeah, and so far I have been, you know, through the supervision and guidance of my supervisors, I managed to actually join the community of supervision, and I've supervised so far about 7 students to completion at honors level, and I've got 2 underway for this year. And I must say, it's all the guidance of HDIs Putlisa and also SAIAB. I would like to thank you very much for this opportunity. Thank you.
Thank you, Dr. Zinzi. So we'll keep the doctor for purposes of inspiration in this talk. Our next speaker is Dr. Ntuthuko Masigane, who started off as one of the first student beneficiaries of the NRF-SAIAB Putlisa program. And interestingly, he is now one of the supervisors under the Putlisa program. So he's had a remarkable journey from being a student and a beneficiary of the program to now being a supervisor. Dr. Masigane will share his success story. Also, he happens to be my PhD supervisor, so I'm quite eager to hear about his background and his story. Over to you, Dr. Masigane.
Thank you, Sazi. So for this session, I'm going to try and answer a question of how do you, as an HCD program like HDIs Putlisa, develop and support a marine scientist from a rural or disadvantaged background. And I hope that some stories and examples will come through. So obviously I'm a lecturer, so a lot of slides, sorry. So I joined SION in 2007. After completing my honors, there was a call for interns in Grahamstown for a SION L1 node. This was recently established. And God mentioned that HDIs Putlisa comes from 2007 effort. So it began as a NASUTUS program that was run by Angus as a node manager back then, and he approached Transnet to fund 2 interns. And at the time we were selected, myself and another colleague, Umandisa Kobuka, who was from University of Fort Hare. So we started in 2007 as interns, and we were later joined by Ulus Bodani. So it started with a student selection, and the way the program was designed by Angus was to target students from historically disadvantaged institutions, bring them into the marine science space to try and address transformation issues. And initially what he did was to provide soft skills that he thinks were relevant to up and coming marine scientists from these programs. I mean, skills like swimming, you can think of them as not important, but for students that come from inland and rurals, that only goes to the beach in December times and stand by the shore and look at the waves. I mean, swimming skills are very important. So such skills were one of the first things that were built into an HCD program. That was the NASUTUS program. And also he made sure that we are involved in a lot of engagement in outreach programs such as Science Week, National Biosciences Month, science camps. And in one of the slides, you see him, myself, and Kububa that were involved in one of those programs. What it does is it opens your eyes into the whole discipline of marine sciences and how it fits in and where you can make a career and connections. Also, some of the skill sets that we're exposed to is working on equipment or the technical skills. So as interns, we're also enrolled into various institutions. So for my MSc, I was enrolled at Nelson Mandela University, supervised by Prof. Chris Woolridge. Luzulu was supervised by Prof. Len Foneman for his MSc on zooplankton, and our research area was Algoa Bay. The nice thing about this is that we were sort of aligned with one of the leading scientists in a particular discipline, because I was looking at marine macrobenthos in that perspective. But most importantly, what worked, which I think was crucial in this HCD program, was to provide ongoing support in all sorts of aspects. So in terms of academic work, the support that we got was getting mentors to go along with us in terms of developing as scientists. So you see, my mentor was Sean Dazel, who's still based in Sunderland, not in Kadeha. He was doing PhD at the time and was doing MSc, had very good relationships with him. In fact, I am now a co-PI on an NRF-funded project with Sean that's looking work as Algoa Bay. Luzo was mentored by Kim Bennett, who's now based in Oregon State University. And Mandisa was mentored by Prof. Nicola James, who is still at SAIAB. So it was quite useful because you had someone you can talk to every step of the way of your academic development, and whichever issues we had, you were the first, or she was the first person to speak to and get advice and all those things. And what worked is that we had like a full year trying to develop our MSc, and you don't usually get so much time to develop one research project. But if you've got funding, it works very well, because then you learn a lot of things along the way. And by the time you implement your research, you know exactly what you're supposed to do. And one of the other things that was provided, like Uzin has mentioned, is inspiration. So going to conferences, having your supervisors, your mentors expose you to some of the leading scientists, like Prof. Brian Allanson. I remember I met him when we were doing a reserve determination study, looking at hot brag and Neisner estuaries. So a part of the training students in that program. And we also went to conferences. My first conference was also SAMS in Cape Town. So another thing that you do if you develop a scientist is to have fun with them if possible, or allow to have a little bit of fun, so they know that you can live within this chosen area of research or career. So as you can see, I was singing with one of the entertainers in that SAMS conference, a little bit of fun there. So after my MSc from NMU, I went for a PhD. It's UKZN, supervised by Randy Newman at CSIR and Prof. Ursula Shala. It's UKZN, and I was lucky enough after graduation to be employed at Forte. So when I was employed at Forte, I remember I was visited by Angus, who was then at SAIAB as a director, and he came with Gath to discuss with me the HDIs Pushes Up program and how it has grown, and to invite me to be part of that program. So obviously I jumped on board because I came from this route, and now I'm doing a lot of research. So that HCD program has developed me into a youngish scientist, and now I'm able to score funding, attract funding, and be involved in collaborative work like an ESSRP This was completed last year with Prof. Shala as a main PI. And I'm also invited into steering committee meetings by WRC or review panels by NRF. So one of the things you let your scientists grow and support them all the way up until you get the rewards of those. And lastly, I think as beneficiaries of such programs, we need to pay it forward in different ways with the others. So obviously I'm in academics, so I do a lot of supervision. So in my 8 years of academia, I've supervised 4 MSc students, currently supervising 4 MSc students and 2 PhDs. I've mentored people like Dr. Madonna Vezi, who is now a lab supervisor at Rainwater, KD under the DST NRF internship, who got her MSc in Spain. There's quite a lot of work that we do in terms of collaborative research with UKZN, CSIR, and other teams. So I think that's how you develop scientists, particularly within the framework of HCD programs. Thank you.
I'd like to thank the 2 speakers. I think that the 2 narratives that you guys have sort of shared with us are a standing example of, or they are testament of the numerous success stories that have come out from the impact of this program and of the achievements in supporting and nurturing young, upcoming marine science researchers. So if there are burning questions to the 2 speakers that we just had, I will allow 1 or 2 questions. I see Smusiso Mukabela, your hand is up. Do you want to engage the 2 speakers?
Sorry, Sazi, I don't have any question. I don't know what's wrong with my laptop, sorry.
Oh, okay. Is there any other burning question or feedback or engagement? Okay, if none, I'd then like to hand over this program to Mr. Zipotanda, who is the administrative assistant for the ASAP Putlisa program at NRF-SAIAB. Zipo has been working on the Putlisa program for over a decade now and has seen many of the program students steadily establishing themselves as astute researchers within the science community. I don't think there's anybody who's in this program who hasn't interacted that doesn't know Mr. Zipotanda. Zipo, please take over. Thank you.
Thanks, Sazi. Indeed, it has been over a decade now that I've joined the NRF-SAIAB and I've been working sorry, with Daragath on the program as the administrator for the program. My work for this session, I think Francesca has paved the way in terms of highlighting the key points that the speakers have alluded when they're giving a presentation based on what the program is offering and what can be accessed through the JMLs. And also I want to emphasize on what Daragath alluded in his talk when he was talking first in terms of trying to secure funding for the next phase. In terms of, so if we keep on using these labs, then we can go to the funders and try to knock on their doors and say that there is a success stories behind all these JMLs, and also it's giving back to South Africa and it's transforming marine sciences within South Africa. And in that, I think they want to see transformation happening in those streams, because we cannot shy away that marine sciences was white dominated, but now with all the transformation that is happening around the country, they can see that indeed also some other people from other different skin colors are able to transition themselves into marine sciences. With all that being said, colleagues, I would like to thank all our panelists on giving us their presentation. And hopefully, as they have said as well, that their doors are open for collaboration I think we must push more on collaboration, because with that we can achieve more in terms of collaborating in all these 4 JMLs across the country. And also with that, thank you very much, colleagues, and thank you, Sazi, for giving me this opportunity to say my thanks, and thanks to Lucky for organizing this session. I think it's one of the best sessions that we had, and also hoping that from this session we'll be able to utilize the resources that are available through the JMLs and also through the HDIs program, as well as the SAIAB facilities that are available for anyone to use. Thank you very much, Sazi. I think my job is done now. Thank you.
All right, what's left of my job is to also thank everyone and say goodbye. This is the end of our program. I'm hoping that, as Zippo has mentioned, that there's going to be further interaction beyond this platform. Thank you so much. Thanks, everyone.
Thanks, Sazi. Oh yes, I would like also to thank all the attendees of this program. I think without them, we wouldn't have such a beautiful program. Thank you very much, colleagues.
Thank you, everyone.
Thanks, everyone.
Thank you.
The panelists want to remain behind just for a short debrief. If you still have time, 5 minutes, please do. Thank you.